Hello, my name is Emily Celayos, and this is my research project. I worked in collaboration with Jesse Kong, who is a student at Stanford, our PI, who is Tadashi Fukami, and Karu Tsuji from Kobe University in Japan. Our project focused on nectar microbes, specifically yeast and bacteria, but our data analysis was centered on yeast, specifically Machinicorbia. Machinicorbia is a nectar specialist yeast, and a nectar specialist yeast is a type of yeast that is particularly adapted to live in environments with high concentrations of sugar, such as those found in nectar. Our samples were collected from the spotted bellflower shown here. During our stay in Ijima, we noticed that it was a rainy season. However, the upside down bell shape of the spotted bellflower made it very ideal for nectar sampling, which is why we chose to use this flower for our project. Again, during our stay in Ijima, there were two things that we noticed that sparked curiosity. That was the environment in which the flower grew in and the rain. The spotted bellflower is actually known to flourish during the rainy season, which made us wonder if there was a potential influence of the weather on microbe growth in nectar. The spotted bellflower is also known to grow on forest sides. However, we found some near bamboo forest or simply between crop fields. This made us wonder if there was any correlation between landscape features and Machinicorbia yeast growth. So those were our two objectives, and we hope that by looking at these two factors, we find a correlation which would help us better understand plant pollinator interactions through microbe growth patterns in nectar and their potential impacts on the dynamics of the surrounding ecosystem. So to start off, we collected samples in Ijima in the Nagano Prefecture of Japan. We collected from 20 sites shown here. And nectar extraction occurred from this little pocket surrounding the base of the stigma, shown here. So after the nectar extraction, we spread these onto petri dishes for microbe growth, and then we noted down the location in which we collected the samples and the weather on the day the sample was collected. There were four categories for the weather conditions, that being sunny, sunny, cloudy, cloudy, and rainy. After samples were collected, we went back to the lab at Kobe University and did a substreak on all interested samples. All interested samples were the ones that looked like Machinicorbia yeast. We then did DNA extraction and PCR and sent them for DNA sequencing. To look at the correlation between landscape features and microbe growth, we first created a 20-meter radius circle around the flowers. In ArcGIS, we then overlaid dynamic world classification data and then calculated the percentage of land covered through the pixel identification, which we then used to generate pie charts. As we see here, and as we expected, we saw a greater percentage of trees in the 20-meter radius in most sites. So for our data analysis, we focused on this forest cover and the tree percentage. Because of the large amounts of growth, we worked with log numbers. So as we see here, we can see that among the four weather conditions in which we sampled, Machinicorbia-like CFU appeared to be the highest during the rainy season, or sorry, the rainy weather. So we were able to deduce that the weather had some type of influence on microbe growth. For our analysis of the correlation between forest cover and Machinicorbia-like CFU and nectar, we initially found there to be no correlation. However, seeing the influence of weather on microbe growth led us to think that maybe we could see a different relationship if we were to further assess this influence of the weather conditions. So we did a subset of data analysis, separating the data gathered from samples collected during non-rainy weather, so excluding rainy weather, and then only the data collected during rainy weather. So rainy weather only. They seem to make a difference, as we can see in this graph, including only rainy weather. There appears to be no correlation. However, when we exclude data from the rainy weather, there appears to be a positive and significant correlation. So as I mentioned before, we focus on Machinicorbia yeast, and we also wanted to take a look at the relationship between the tree percentage and the percentage of yeast in nectar at each site that visually appear like Machinicorbia. Initially, again, we noticed no correlation, but we decided to do the subset again of the analysis, excluding rainy weather and then only considering data with rainy weather conditions. We saw the same pattern where with the rainy weather, there appears to be no correlation. But then excluding this data, we begin to see a significant and positive correlation between the tree percentage and the percentage of Machinicolia like CFU. So we concluded that rainy weather does appear to enhance yeast growth in this specific flower, and that there is a significant correlation between forest cover and yeast abundance, but only when sunny or cloudy, not when rainy. As I mentioned, before we focused on Machinicolia nectar specialist yeast, and we greatly depended on visual aid. So here we have our prediction of whether or not a yeast visually appeared to be Machinicolia, and then we only sent those that did appear to be for DNA sequencing. Through DNA sequencing, we confirmed that 95% of the selected samples were in fact Machinicolia. So it was very helpful to see that we could potentially visually identify this yeast. In the future, we hope to assess similar relationships in other species of yeast from the samples and perhaps in other flowers too, as well as assess the effect of the weather conditions on nectar microbes in a controlled environment to potentially find direct causation. For further acknowledgements, we want to thank the Suji Lab, RESERP, Japan Society for the Promotion of Science, SDSS Discover Grants for sponsoring us. Thank you to Hidenori Deto and Tadashi Miyashita from University of Tokyo, Hongo Nosan, Ijima Town, and their local farmers for their cooperation with our fieldwork, as well as a very special thanks to all of the members of the Suji Lab for their help both in and out of the lab. Me and Jessie are very grateful, and yes, this is my project for the summer, and thank you for listening.